Think this is awkward? Try being proline. This amino acid side chain folds back on itself to form this weird ring. But if it's next to the loosey-goosey glycine, this allows them to work as a team to build triple helixes of collagen protein that make our skin strong. But to make that, the proline actually has to get modified to have an OH group added. And this is done with an enzyme or a reaction helper that needs vitamin C. And so this is why you need vitamin C or you'll get scurvy.
So we are halfway through 20 days of amino acids, and we are talking about proline. So proline is one of the weirdos, said with love. And we're going to see how it's going to pair up with another weirdo, these weirdos, glycine, to form those collagen structures. And it's really cool. Yeah, I love the weirdos. So why is proline so weird? Well, like all amino acids or protein letters, it has a unique side chain. So it's got the generic part, the amine group, and the carboxyl group. But its amine group actually, its side and this unique part or a side chain or R group, but its unique part, it actually like folds back on itself and reattaches to this nitrogen. And this is going to have consequences in terms of both like the angles it can take and the type of structures it can be. So normally the nitrogens in the charged state, their cationic state, there's like 3 nitrogens. And what you have is the amino acid has a primary amino group, which means that this nitrogen is only attached to one other carbon. Proline is the only one where you have a secondary amino group, which means that the nitrogen is attached to 2 carbons. So you have this nitrogen attached to 2 carbons in this proline. So this is a secondary amine group. So when the amino acids link together to form like through peptide bonds to form long chains that fold up to give you proteins, what's going to happen is that when they link up, they're going to do so like through that amine group. And when you do this with most amino acids, you're still going to have a hydrogen sticking off of that nitrogen. When you do this with a proline, however, you're not going to have a hydrogen there. And so it's not going to be able to participate in hydrogen bonding. So what do I mean by hydrogen bonding? So basically all of these like circles, like the blue circles, the nitrogens, the red circles, the oxygens, the black circles, the carbons, and those gray ones, the hydrogens, those are all representing atoms. And so those are like the smallest units of elements. But they're made up of smaller parts, subatomic particles. There's the positively charged protons hanging out in a dense central nucleus with neutral neutrons, which kind of help glue them together. And they're raining in a cloud of negatively charged electrons. And they can actually share electrons to form bonds, form covalent bonds. So when they're linking up with one another, they're going to be joining pairs of electrons, like kind of merging their electron clouds to form those strong bonds. So these bonds like between the oxygen and the hydrogen, and between all of those bonds between the various atoms in like a proline. But they don't always share fairly. And because the protons are positive and stuck in place, and the electrons are negative and shiftable, when they're sharing pairs of electrons, the electrons can hang out more with one of the atoms than another. So you can kind of think of that electron cloud kind of shifting more towards one of the atoms or versus the other. And this is going to happen if the atoms have different electronegativities, which is basically a description of how well that atom can pull the electron density towards it. And so oxygen is really electronegative, and so is nitrogen. So they can pull the electrons towards them and become partly negative. This is when they're attached to a hydrogen. So the hydrogen, it only has a single proton, so it's really bad at like trying to rein in electrons, because remember, you need that positive pull from the protons to kind of rein things in. And so it's going to really have stuff stolen. And this is going to make it partly positive. And when you have partly negative and partly positive things, you can get them to attract. And so that here, when we're talking about like a hydrogen bond, you're talking about one of these attractions between a hydrogen on an oxygen or a nitrogen. and then a nitrogen or an oxygen with a lone pair of electrons. And so these are like unpaired electrons. This can happen in the peptide backbone because the hydrogen on the nitrogen can act as a hydrogen bond donor. So that's donating the H. And then the oxygens on the backbone can act as a hydrogen bond acceptor. So this will be the partly negative part, and this will be the partly positive part. And so this will allow when you have the peptide backbones, when those amino acids are linked up, they're done in this way where you have these alternating donors and acceptors and donors and acceptors and donators and acceptors. But in the case of proline, you don't have the donator. So you don't have a hydrogen on this nitrogen once it's linked up. And so you don't have the ability to serve as an electron donor, because those hydrogens are going to be like donors. This is going to be attached to a carbon, and you're not going to have a hydrogen to hydrogen bond. And so this is going to restrict the types of structures that it can be found in. And the reason why is that it's those hydrogen bonds of the backbone that are really important for giving a protein its secondary structure. And so we can talk about, when we talk about amino acids or protein structure, so basically you can think of these amino acids, they link up through these peptide bonds. And these peptide bonds are resonance stabilized. So basically there's partial double character throughout here. And so you can't rotate around this peptide bond. You can only rotate on either side. And so you end up kind of having like a chain of planes that you can rotate around. But this then they're going to fold up to form a protein structure to best accommodate all of like the unique side chains or R groups of those amino acids. So we can talk about protein structure at a lot of different levels. So the primary structure is basically just the sequence of amino acids. The secondary structure is going to involve backbone interactions, and this is going to happen through hydrogen bonding. So more in secondary structure in a minute, and then we have tertiary structure, which involves the side chains, and quaternary structure where other peptides are joining in. So all of this is all involving like non-covalent bondings. These are just like attractions. The only place where you have like covalent bonds is if they were like cysteine side chain cross links or whatever. But so your primary structure, when these are all linked up, this is through covalent bonds. Those are the strong bonds where you're actually like merging your clouds. With all of this, it's just like attractions, even though we call this hydrogen bonds. But because you have that regularly alternating like donor acceptor from the hydrogens on the nitrogen and the lone pairs on the oxygen, what happens is you can get these characteristic folds or structural motifs that come because you have this regularly repeating structure. There's common ways that they can best accommodate to take advantage of the hydrogen bonding opportunities. And so the most common motifs you see are like beta strands, which are like these zigzaggy lines, which can team up to form beta pleated sheets, and then like alpha helices. And so this is going to, these are often like connected by flexible regions called loops or coils to give the chain its overall 3D shape, which is this tertiary structure. And so some proteins are made up of multiple polypeptides, and so they also have a quaternary structure, as we'll see with collagen, but in this example, it's only one. So here you can see how like where these bonds are, how these structures form with these hydrogen bonds shown in these like dotted lines. And if you have a proline where you have you're going to have the absence of a donor. You can't, so it's hard to see when it's just like a free amino acid, but when it's linked up, then it's not going to have this hydrogen here. So it's not going to be able to form, it's not going to be able to act as a donor, and so it's not going to be able to participate in these characteristic bonding opportunities that most amino acids can. And so this is going to make it so you won't find it in this sort of places, but where you will find it is because it is kind of like kinky, it kind of it can provide like a structure, like strength to proteins, because it kind of like locks things in place more, as opposed to like being able to rotate a lot. And you'll also find it kind of on the kind of in regions where you need to like turn and that sort of thing. So often between like structural motifs. And it's often preceded by a glycine, as we will see, because the glycine is small and flexible. So although all of them except for proline have this ability to form these like secondary structural elements, they have like the donor and acceptor they need to do this. They don't all have like a strong propensity to do this. And this is going to be in part based on like how bulky or awkward their like side chains are and how that's going to limit their motion. And so how you can rotate the bonds. So when we talk about rotating the bonds, we're not rotating the peptide bond. So the peptide bond, that's like this linking up these amino acids. So it's between like this nitrogen, this carbon, and this oxygen. There's like resonance. So basically they're sharing electrons. So if you have like a single bond, you're showing a pair of electrons. A double bond, you're showing two pairs. When you have resonance, it's kind of like you have partial double bonded character through multiple electrons. So they're kind of like sharing that second electron that you would need, or that second pair of electrons you would need to form the bond. So yeah, this kind of like communal sharing of electrons, and that's going to stabilize it, and it's going to give it double bonded character. When you have a double bond, you can't rotate around a double bond. And so this is going to be planar. So you end up getting kind of like this chain of planes where you can rotate around these various, these 2 points, this like phi, this will get you like the phi and psi angles. And so basically this rotation though is going to be limited in part by your side chain. So they're characteristic like phi and psi angles. So those angles like between the planes that amino acids can take. But some of them, so this is like where you would common for just like a general amino acid where you would expect those angles to be. But you can see that some of them are weird. And so proline is one of the weird ones. So proline and glycine are the two that have like the most different normalness. So proline, its awkwardness is going to limit the angles it can take. And so it's going to have this is showing you like the places where the angles are likely to be. And you can see with proline that it's a lot more restricted. And so this is why like proline could help kind of give strength to a region of a protein by kind of locking things into place. On the other extreme, you have glycine. So glycine, its side chain is just a hydrogen. And so this is going to be really loosey-goosey. And so you can see that glycine has a much broader range of angles it can take. And so these two can team up to make this really great pair. And this is what we see in collagen. So in collagen, you have glycine, proline, and a modified form of proline called hydroxyproline that can actually form these really long chains that can kind of, you get this like glycine, proline, or hydroxyproline, but basically it's like glycine and then a proline and then a proline or a hydroxyproline, and you have this like repeating motif. And that can form these collagen triple helix, which is really cool, and it's going to give the strength to our skin and bones and all of that good stuff. And so in this, you have this, so this hydroxyproline, basically this is a post-translational modification. So by translation is where you're linking up these amino acids into the chain. Post-translational modifications are when you make changes to the amino acids after they've been added. And so only certain amino acids can get post-translationally modified in different ways. So there's like phosphorylation, acetylation, methylation. Proline is subject to hydroxylation. And this is where you add a hydroxyl or this like OH group. It can be added at a couple different places. So there's like 3-hydroxyproline or 4-hydroxyproline, just like different locations where the hydroxyl group is added. So this 4-hydroxyproline is like, there's like 100 times more of this one than this one. So when people say hydroxyproline, they're typically referring to this one. And in order to add that, there's this enzyme, so it's like reaction help speeder upper mediator thing called proline hydroxylase, and it needs vitamin C as a cofactor. So a cofactor is basically like a helper molecule that enzymes can bind to and gives the enzymes like extra different functional groups, so different like chemical arrangement type things that it can use to do things. Sorry, I'm really tired. And so this one, it uses vitamin C. So vitamin C is ascorbic acid. You can see it's this thing. And so what happens is you need vitamin C to do this, and you need the hydroxyproline to make your collagen triple helix. And so with scurvy, you don't have enough vitamin C. You can't make your hydroxyproline, and so your collagen, you have problems with your collagen. Hence, you have bigger problems. And yes. So this is one cool thing, structural thing about proline. And so you can see that the glycine, like preceding the proline, is going to help because the proline's kind of awkwardly can't move much. The glycine can move a lot. And so the glycine is going to help it out. You also see glycine before this weird thing called a cis-proline. And so basically most peptide bonds are almost always in trans. And what this means is that when you have, so in trans, the alpha carbon, so that's the carbon that's going to be attached to the alpha carbon is the one that's like attached to the side chain, and then like the carbonyl on one side and the amine group on the other side. And when you have in trans, the alpha carbon, so I've shown them as like green dots here, those are going to be on opposite sides of the peptide bond. So remember, this peptide bond is like a planar, and the carbon group, the alpha carbons are on opposite sides. And so this ends up you getting like this zigzag backbone. When you have in cis, these hydrogen alpha carbons are going to be on the same side of the peptide bond. And so your backbone's kind of like U-turning. And this is really, really uncomfy for most amino acids. But proline can sometimes form this. So we can call this like a cis non-proline or a cis non-pro. But proline, you can sometimes get cis-prolines because the proline's weird backbone is so weird. This isn't that uncomfy for it. But in order to switch between these two, it involves like actually breaking the peptide, like rotating around a peptide bond. And since that's a double bond, you need an enzyme to help you do this. And this is done with like a peptide prolyl isomerase enzyme, which can convert between them. So these aren't just going to like spontaneously like switch things up, but like, so after they're put in the trans way, then this peptide prolyl isomerase can switch them to cis. And so, but this is rare. And so basically about 5% of prolines are cis, so pretty rare. But really more rare is that if you have a non-proline cis, so only about like 0.03%. So with the cis and with the cis prolines, they're often preceded by a glycine, such as because glycine is so like flexible and stuff, or by an aromatic amino acid. And the non-proline cis bonds are like super duper rare, and if they do occur, they often have like occurring in like an active site where there's like some functional some functional purpose for having it like that. And so this comes up in crystallography. So X-ray crystallography is this technique where we can like figure out like the atomic structure or the near atomic structure of proteins. So basically how all the atoms are like orienting like where they are in 3D space. And so it works by shooting X-rays at a crystallized protein. So basically in a crystal, all the protein copies are like oriented the same way in this orderly lattice, and you shine X-rays at them, and the X-rays diffract, so they kind of get like scattered by the atoms of the protein. And then those scattered X-rays are going to interact, and they're going to interact constructively, like building together, or destructively, like canceling each other out. End up, you get this like pattern of spots called a diffraction pattern that you can work back from. To get this electron density map showing us where the electrons are, because that's what's actually going to be doing the scattering, not like the center of the atoms, it's the electrons around them. So you end up getting this kind of like meshy thing, and then you build an atomic model into the map. So the atomic model is what you're looking at when you see those like pretty structures. But when you're actually doing figuring out the structure, you're working with this meshy thing. And sometimes if you have low resolution data, this meshy is going to be really meshy. And if you have high resolution data, then it's easier to figure out where the atoms actually are. And when you have lower resolution data, then it can be really hard to accurately place the various atoms. And so sometimes if it's too bad, we don't even try to model the region. But sometimes if you're trying to model things in, you might model in things inaccurately. And that's where this mole probity software can really help make sure that you're not doing that. And so this paper on mole probity, was developed by Jane Richardson and her husband David. They have a lab at Duke. And so Jane Richardson's really awesome, and she was the crystallographer who invented like the ribbon diagram representation of protein structures. And they also developed this mole probity software, which is going to help point out potential problems in structures. So it's actually integrated in with Phoenix, which is like a software that you can use when you're doing the modeling and that sort of thing. And so this integration is really nice because you can like see the problems as they're happening, and you can get like a mole probity validation report showing you where like the errors are and stuff. So this is like the mole probity software, and you can see that they like point out this software is used to like validate crystal structures and stuff. So basically those pretty pictures of the proteins that are got using X-ray crystallography. And so sometimes you might put things like wrong, like the backbone is weird or stuff. And mole probity can help point out the errors by like kind of like flagging them visually. And so they flag the cis non-proline bonds. And this has had a really good effect on helping make the crystal structure more like realistic and better. And so basically you can see that so this is the percent of the PDB deposits with more than 30-fold too many cis non-prolines by year. And so the PDB is the protein data bank where all of these like crystal structures are deposited and stuff. More on that in another post. So you can see that like in the 1990s, like it wasn't that big of an issue. Then they were super high for 10 years, like between like 1.5 up to 1.5 like times percent of the deposits had way too many cis non-prolines. And then with the introduction of mole probity, it's drastically decreasing. So basically people kind of like, oh, cis-proline non-prolines can exist, that remember that like 0.03%, so they can exist, but they can technically have, and they tend to have like big purposefully exist. But people were like, oh, we can just use a cis non-proline if it helps us make our structure make sense. Like if your structure is kind of blobby, it can be kind of easy to kind of fit cis non-proline into the blob, which is a no-no. But so basically, and then if your software wasn't pointing it out to you, then you might leave it in there. But with the mole probity, it's like pointing out like, hey, this is probably wrong. And so then you can fix it. So size-wise, proline's not that big, but it is the most restricted. So some of these are restricted in their motion because they're really big. Proline's restricted because it has that weird loopy side chain. So although it's not very hydrophilic, so water doesn't like it that much, it's still often found on surfaces of proteins because it's really good for kind of like forming turns and that sort of thing, connecting the stronger secondary structural motifs. Proline is non-essential in the dietary sense, meaning that our bodies can make it ourselves. We don't need to get it directly through our food. Or pre-made through our food. And so it can actually be made from glutamate, the amino acid. It's another amino acid we'll look at later. So some of these steps are helped along with enzymes. So this glutamate can be enzymatically transformed into glutamate 5-semialdehyde. So we see that we've lost this oxygen. Then this can like spontaneously ringify to give you this like pyrroline 5 carboxylate. And then this can get reduced. So here you see like a double bond, and here you have a single bond to give you proline. You can also make stuff from proline. So proline is glucogenic, so it can be used to make sugar. And so basically proline can get converted into alpha ketoglutarate. And this can enter the tricarboxylic acid cycle, like a Krebs cycle or citric acid cycle. And then this can be used to make oxaloacetate, which can be used to make glucose. So we call it glucogenic. So history-wise, it was first isolated in 1900 by Richard Willstatter, and its name comes from pyrrolidine, which is the chemical name for its side chain. And so I hope this makes you better appreciate the weirdo proline.